All right. So why we study gene expression? We talked about how gene regulation is really very important in making the different cell types and how they function in our bodies. So we spent a lot of time about gene regulation at the transcriptional level, at the post-transcriptional level. And now one of the things we can do to understand how our bodies work, how different animals work, is we can look at their at the gene expression levels of different genes in their different parts of their bodies upon different type of environmental signals, stimuli, etc. Right? So the take-home message from all the semester has been that different types of cells express different genes, making specific proteins and functional products for specific cell structure and function. So then to understand the differences between cell types or between two states, such as in a given cell type, normal versus disease state, one of the things that we can do is we can study the differences in gene expression. So we talked about genomics. That's the structure, the study of the sequence, structure, evolution, and function of the genomes. Now we're going to add another omics, which is the transcriptomics. Okay, so the transcriptome is basically the set of all the RNA molecules in a cell or a tissue or organism. And the transcriptome between a nerve and a muscle cell will help us understand how these two cells' structure and function are different, right? That makes sense. All right, and then one of the examples is that in the nerve cells you find genes that are expressed, such as acetylcholinesterase and tau protein, and in muscle cells other genes are expressed, such as acetylcholine receptor and tropomyosin, right? And those are just two examples. But you expect that a lot of, many different genes would be differentially expressed. Differentially expressed means expressed differently in two samples that you're comparing. All right, so far so good. You already seen some techniques in which you can measure gene expression. All of these techniques were low throughput. That means you could study only few samples or few genes at a time. And one of the examples that you've seen were the northern blot analysis that looked at the, for example, beta globin mRNA. And the way that this works is you isolate RNA, run it on an agarose gel, transfer all the RNA to a nitrocellulose membrane, which is kind of a nylon membrane. And then you make a radioactive probe that is against, that it has a sequence that's complementary to the gene that you want to study. In this case, it's beta globin. And you can see the signal here, that's the beta globin. And the more signal you see, the more of that mRNA was present in your sample. You also saw in situ hybridization, which is another technique that depends on complementary annealing of your probe that is labeled differently, and you can study again one gene at a time. And here, what you can see here is you load the whole total RNA on a gel, so you have absolutely no spatial information. That means you don't know where in your sample that gene was expressed. The in situ, you have the spatial information, right? That means where is it expressed? It's expressed in the head and the tail. That looks like the notochord, etc. Does that make sense? Both of these techniques are low throughput. That means you can look at a few genes at a time. If you wanted to look at the transcriptome, you would have to repeat this northern plot 2,500 times, right? I mean, 2,500, oh my God, it's too early. 25,000 times to look at all the genes in the genome. So these are not very good. We need a high throughput method to measure the transcriptome of a given sample. All right, so here are some of the techniques that we can use. We're going to go and talk about microarrays because some of the themes and principles will apply to many other techniques. We're going to talk briefly about single RNA-seq and very briefly about single-cell RNA-seq. All right. The microarrays have exactly the same principle as a Northern blot or in situ. It depends on the complementarity of a given probe and how much it can hybridize to your sample. Okay? So a DNA microarray is a collection of microscopic DNA spots attached to a solid surface. So here's a cartoon version of this coming right out of, is it in your textbook? I don't remember. Anyway, what it is it's a glass slide. And this glass slide has what we call probes. These probes are short DNA molecules. These can vary between 50 base pairs to a couple of kb long. All right, and they are synthesized. So you synthesize them, and they each have a sequence to a corresponding gene that you want to analyze. So this could be the sequence of that's complementary to beta globin gene, right? And you can also spot another probe that would be complementary to a second gene, and a third gene, etc., etc. So however probes can you fit on a surface of a glass slide will determine how many genes you can analyze at a time. All right, so after you prepare this glass slide with all the probes, which is the DNA microarray, what you do is you isolate the RNA from your sample that you want to analyze, and then you create probes basically using that RNA sample, right? What you do is you take that RNA and you convert it into a cDNA, and that cDNA is going to be fluorescently labeled. All right. Let me see here. I had a question there. I have the question in the wrong slide. Okay. How can you introduce a fluorescent label into a cDNA from an RNA sample? You isolated the RNA. You're going to make a cDNA, but you want to make sure that cDNA is going to have fluorescently labeled nucleotides. So that cDNA is going to be labeled. How can you do that? Any ideas?
Would you add like a fluorescent tag to the primer?
Well, if you used, that's one way to do it. To make cDNA, you can use primers, right? So your RNA looks like this. Let's say it's a mRNA. It has a poly A tail. So if you use the poly T oligonucleotide to make the cDNA, you could put a fluorescent label over here, right? That's not very common, actually. We tend to, because what happens is that because the reverse transcriptase is a crappy enzyme, it falls off after it starts making the cDNA. So you always capture the 3' end of the gene much, much better than the 5'. So what we tend to do is we use short hexamers. So these are random sequences that have 6 nucleotides. That's why it's... And these short hexamers serve as the primer for the reverse transcriptase to make the cDNA. Okay? And one thing that you can do is you can have these short hexamers fluorescently tagged, or what you can do is when the cDNA is being made, you can add the DNTPs that are tagged with a fluorescence tag. Sorry. So there are at least 3 ways in which you can create fluorescently labeled cDNA, right? You can have fluorescently labeled oligo-DT. You can have a fluorescently labeled hexamers. You can add fluorescently labeled DNTPs. So the reverse transcriptase, when it's making the DNA, it's going to incorporate those nucleotides. So at the end, you'll have labeled cDNA, complementary to the mRNA samples that you have in the reaction. Does this make clear? Is this clear? Questions about this? It's going to be important, right? So if you don't get this, you'll not get the next thing. Got it? All right. So then you have the labeled nucleic acids, right? And the labeled nucleic acids, you're going to take them and you're going to hybridize them to all the probes that are fixed onto the glass slide. So if your sample, fluorescently labeled sample, in this case, what you would call your sample, hybridizes to a probe, that spot on the glass slide is going to have a fluorescence signal, right? So if you have a scanner, then you can scan that slide and say, that probe that I printed there, that is against beta globin, is showing a fluorescence signal, telling you that you had beta globin RNAs that you isolated and labeled and it hybridized, right? All right. And so if there's another spot that doesn't show any fluorescence signal, you know that you didn't have that mRNA corresponding to that probe, that spot. All right, so here's kind of the working principle, just summarize. You isolate total RNA, you incorporate fluorescent nucleotides while converting to cDNA. The synthesized cDNA number then will be proportional to the initial RNA number, right? So if your sample, there was one gene that was expressed thousands of copies, you have thousands of copies of that mRNA corresponding to that gene. And if a gene is expressed at a very low level in 1 or 2 copies, the number of cDNA you're going to get out of those is going to be low, right? So it's going to be proportional to the number of RNA. So a gene that's highly expressed will have many mRNA, and a gene that's lowly expressed will have fewer RNA. So in this toy example where I'm looking at only 2 genes, gene 1 is expressed a lot, gene 2 is expressed at low levels. You hybridize it to the glass slides. All the gene 1 probes at one spot on the slide is going to be is going to have some signal, and all the gene 2 probes that are spotted on the other spot in the slide is going to have fluorescence signal. So which one of these spots will have more fluorescence signal? This spot here, gene 1 probes or gene 2 probes? Gene 1, yeah? That's very simple. All right, so that's the principle. You have more RNA, you're going to get more signal because there's more cDNA. How do we do this experiment? Usually using DNA microarrays, we compare 2 samples against each other. That will allow us to determine genes that are differentially expressed between 2 samples. Muscles versus neurons, cancer versus normal, okay? Time point 0, time point 2 hours, whatever. Whatever you want to measure, that is when you want to measure the differences between 2 samples. So how do you do that? So microarrays are used to make direct comparison between 2 different RNA samples. So it provides a readout of the relative differences in abundance. Okay, so what you do here is this. You have a cell type A that you're going to compare to cell type B. You extract the mRNA, so you have mRNA from both, and then you make the cDNA. And when you're making the cDNA, you use 2 different fluorescents in this. Okay? So in this one, for example, you can use a fluorescent nucleotide that is going to give the cDNA a color of green. And in this one, you're going to use the fluorescence that gives the cDNA color of red. So then you take these 2 samples, you can mix them up and hybridize them both to the DNA microarrays. And then when you look at the glass slides, some of the spots, so each of these spots corresponds to 1 gene, right? And some of these spots are going to be more green, and some of these spots are going to be more red, and some of these spots are going to be yellow, right? So if this spot is green, for that gene, is this gene expressed more in cell type A or cell type B? It's more green. A, right? And conversely, if it's red, it's going to be B, and if it's yellow, it's about the same. But we want to quantify this data, right? And how do you quantify it? Before I get to quantify, here's the example actually from your textbook. what you're looking at here is you're looking at the differentially expressed genes upon serum addition in fibroblasts. So you are going to look at the genes that respond to serum addition. So what you do is you grow your fibroblasts in two different dishes. In one, you don't add any serum. In the other one, you add lots of serum. So this is your no treatment control, and this is your treatment. And you isolate total RNA. You incorporate the green dye, which is Cy3 here, and the red dye, which is Cy5, and you combine this cDNA. You mix them up and then hybridize it to the microarray. And each spot is corresponding to a gene. And in this example, there are 8,600 genes on this microarray. And then you can measure the level of fluorescence you get from each spot. Okay. So the readout is this. For each spot, your scanner is going to tell you how much fluorescence it's going to see from CY3, how much fluorescence it's going to see from CY5. That's the fluorescence from one sample, fluorescence from the other sample. So to look at the differential expression, what you can do is you can express this quantitative data as a ratio, right? So you can take the ratio of one signal, CY5, so that's the amount proportional to amount of RNA in that sample, compared to the other one. So you can see here that for this gene, when you take a ratio, that gene is expressed 50 times higher in the treatment sample compared to no treatment sample. Okay? And in this example, when you take a ratio, you see that that gene is expressed 30 times lower in the treatment sample compared to the no treatment sample.
Is there a way of measuring like absolute values of like how much mRNA is expressed, or is it just relative?
This is mostly relative. There are techniques in which people have tried to develop absolute measurements. It requires a lot of controls in which you include synthesized RNAs that you know exactly how much they are. So you can you can use them as a way to standardize your signal against theirs and try to come up with absolute numbers. It's more difficult to measure absolute numbers than relative numbers because at every molecular biology step there is a level of efficiency that might change from experiment to experiment to experiment. So relative is always easy, but absolute requires some kind of standardization. Okay. Any other questions? All right. So one thing that you might notice here is that when you take a ratio of a higher number divided by a smaller number, you get numbers that are high, so that's going to be 50. But if you take a ratio of a small number to a bigger number, your ratios are going to be all squished between 0 and 1, right? The other numbers are going to be very, very high. All the smaller numbers are going to be squished. So the range is going to look wider when comparing fold increase. That means in your treatment when you have more of something, but it's going to be squished when you're looking for fold decrease. So when we want to visualize this and compare it, what we do is we take a logarithm of this ratio. So when you take a log 2 of this ratio, what you can see here is that you can convert those numbers between 0 and 1 to a wider range, right? So now the log 2 of 50 is 5.64 and 0.03 is minus 4.91. So when you look at then what you can do is you can take these numbers, which is a measure of how much change there is, and you can convert it to some pseudo color, whatever you want. You can make this more red. That is, if a gene is increased upon your treatment, you can give a red color to that when you're trying to visualize it, and a green color. So red and green were chosen just out of the first people who developed microarrays, which were Pat Brown and his friends. They chose red and green, and that has been the tradition for a very long time. But we know that 10% of the males in this room actually can't see anything, right, including my husband. When he looks at microarray data, he's like, I don't know what's going on, because he's colorblind, right? So people now express these data in yellow and blue, which is much more helpful. But in this example, it's red and green. Okay, so how do we actually visualize this data and analyze this data? So what you have here is that you have the glass slide. Each of these spots is a gene, right? And you have a measurement of fold change in the mRNA level between two samples, and each of these measurements are quantified as a number, but you can also express it as a color. So what you can do is you can organize this data into a matrix. You can take each of these spots and put each gene in a row or a column. In this example, it's in a row, okay? So each gene is put in a row. And then you have multiple microarrays, right? You have, for example, experimental replicates, because doing one experiment once is no good, right? It needs to be reproducible. Or it could be different samples, right? It could be treatment of patient 1, treatment of patient 2, etc., etc. So what you can do is you can put all the genes in the rows and all the samples in a column, and then you can color code it. In this example, now we moved away from red and green. Because everything is arbitrary, you can choose whatever color that you want. So in this toy example, we removed all the intensity of the colors and said, if it is higher in the treatment sample, make it purple. If it is lower in the treatment sample, make it color it green. Okay? So in this sample 1, the gene 1 went down, gene 2 went up, gene 3 went down and down and up and down and down and up. So these were what we call upregulated. So upon treatment, their expression went up. These were downregulated. Upon treatment, their expression went down. So you put all the data like this, but can you see any patterns from this? It's very difficult, right? And in this toy example, I only have 8 genes. In real examples, we have thousands of genes. So it's very difficult to see any patterns from this. And if you don't see patterns, you can't interpret the data. Pattern recognition is basically how we learn, right? So what can we do? Imagine this is a situation where you want to see this picture, right? You have this beautiful picture, but this picture went through a paper shredder, right? And each of these rows is a gene. So you went through the paper shredder, and when you put it back together, they were all jumbled. You don't see any pattern. What you want to do is you want to take this whole jumbled data set and then try to make it so that you can see the underlying patterns that this data set is actually telling you, right? And that's a little bit difficult. There are different ways to do this. One of the ways that people have done this is what is called hierarchical clustering, okay? So this is actually Mike Eisen's work, where he wrote one of the very first algorithms to do this, and he applied his algorithm to this picture just to show how it actually works. So the principle is this. What you do is you look at 2 columns and then look at the pattern of their gene's expression and take the 2 columns that are most similar to each other and place them next to each other. So here you can see that the sample 1 looks very much similar to sample 6, right? Same genes are going up and same genes are going down. What you do is you take that sample 6 and now put it right next to sample 1. So you go to the next one, you look for similar. You look at this column and say which of the other columns looks most similar to this, and you grab sample 5 and put that to sample 2, and you do the same for 3 and 7. And now you have some kind of clustering, right? So this is hierarchical clustering. You look for most similar samples and you put them right next to each other. And then you can create these relations. That means this 1 and 6 are more similar to each other. 2 and 5 are more similar to each other. 1, 6, 2, 5 more similar to each other than 4, 3, and 7. Does that make sense? So that's how you do your clustering. And now once you do this clustering, you can see patterns in the data. And I'm going to show you a real example from this. So in this example, what the researchers have done is looked at all of the genes that respond to the serum addition, right? It's our example where fibroblasts were treated with serum, and we're going to look at which genes went up and which genes went down. For genes that went up, they were color-coded red. Genes that went down were color-coded green. And genes that didn't change were color-coded black, okay? And so instead of having the genes in the rows, these researchers actually organized the data such that every column is a gene, okay? So you have thousands of genes here. Every column is a gene. And on the y-axis, their samples were a time course experiment. That is, what they did is for every, after serum addition, they took a sample out at 0 minutes, 10 minutes, 20 minutes, 30 minutes, etc., etc. So they can follow the progression of the gene expression response to serum addition over time, right? So that's another way you can use microarrays. So we're looking at the expression of every gene in each column across time in each row. So this is the beginning of the serum addition. This is the end of the serum addition. Now, all the genes are clustered, and that's the clustergram on top shown here. And what you can realize is that some group of genes respond to the serum addition similarly. So in this group A, what you can see, so let's say, in group D, so this is a group of genes that do not respond very much to serum addition very early, but then their expression goes up, right? And then towards the end of the treatment, their expression goes down. There's another group of genes that respond to the serum addition with some delay, but their expression seems to be pretty similar towards the end, although there's some variability. There are some genes, as soon as you add the serum, their expression starts going down, but then they recover in later, right? So you can see groups of genes being regulated similarly, right? Okay. What's happening to group A after serum addition? Just looking at these colors, can you tell me, just like I told you for the other groups, what's happening to the expression of these genes? Yes, exactly. At the earlier time points, you don't see very much change to their expression, but then their expression goes down with time. All right. What can you do with that kind of cluster information? You can do a whole bunch of stuff. I'm just going to show you a few examples, okay? For example, what can you do? These researchers, what they did is they said, okay, we want to know, we have a lot of breast cancer patients coming in our clinic. We actually take out the breast cancer, but then some of these patients get metastases within 5 years, and they get very, very sick. And some of these patients walk out, and 5 years later, they're completely cancer-free. What is the difference? Can we actually tell what the difference is going to be at the time of the surgery, so that if we could predict which patients were going to develop a lot of metastases, we could treat them with more aggressive therapies, right? You could give them chemotherapy, etc., etc. But if you knew a group of people who are going to just survive and they're not going to care, maybe you don't want to put them through very invasive or horrible therapies, right? So what can we do? So they designed this experiment. What they did is they took samples from tumor samples from a bunch of women that went through the surgery, and they did a microarray analysis, and then they followed these people 5 years later. And then what they knew was that some women had a good prognosis, they recovered, and then some women had bad, right? So then they knew which sample was coming from which person. So what then they did is they took the data and they clustered it. So this is every row is a person, right? So it is a sample, and every column is a gene that they looked at. And these are the genes that seem to be most different. And what you can see is that for a group of patients, right? So this group here, a bunch of genes that are highly expressed and a bunch of genes that are lowly expressed. There's another group here which shows a kind of a similar pattern, but the gene expression is much more variable than this one, right? So they clustered in another group. And you can look at a third group where a bunch of genes are much more highly expressed, but the same genes that were highly expressed in group 1 are actually lowly expressed. And then in group 4, there seems to be absolutely no pattern. Things seem to be very, very random, right? So the data by itself hierarchically clustered these patients to the 4 groups. And then they said, let's look under each group how many of these patients showed a bad prognosis 5 years later, right? And they found that those patients that showed this gene expression profile, none of them were showing a bad prognosis. None of them showed metastasis. In group 2, 6 out of 21 actually showed it. In group 3, 18 out of 24. And in group 4, 10 out of 13, right? So this actually told the doctors that maybe we can use microarrays to predict if a patient is going to have a bad prognosis or not. So, for example, if you were that doctor and a patient came to you, you removed the tumor, and then you did an expression profile, and the patient's profile clustered with group 1, would you do a very invasive hard therapy on that person? Probably not, right? You would say, come back 5 years later and let's take a look at you again, right? But if that person was in group 4, what would you say? That person is probably going to have a high chance of developing metastases. Maybe you need to come 6 months later, a year later, and maybe we should take a look at you again, right? So that's how you can use microarrays. All right. Microarrays can be used when other techniques are not very good in diagnosing what kind of cancer a person might have. So for a lot of doctors, one of the ways in which you could tell what kind of cancer a person has is you take out the tissue, you send it to histology lab, and then they look at these tissues and they tell you, okay, these are central nervous system tumors. They're coming from this type of neuronal cells or that type of neuronal cells or et cetera, right? But one of the problems is sometimes they are not able to tell this difference by just looking at the picture. These cells look very similar to these people. It also looks very similar to me because I'm not a histologist. But one thing that people could do was use microarrays to differentiate the different types of cancer cells. And what they did here is that they have a bunch of genes here. So each row is a gene. And each column is a cancer sample where they know what kind of cancer they're from. So this is from medulloblastoma, this is from glia, and et cetera, et cetera. So they look at the expression profiles in each samples and cluster it. And what you can see is that medulloblastoma samples cluster together and away from these samples, et cetera, et cetera. And what you can recognize is there are some genes that are distinctly high in one type of tumor and low in another type of tumor, right? So if you could then isolate these genes and determine which genes have the most predictive power to distinguish these two classes, then you can do a much more simpler a PCR test or something like for diagnostic labs to be able to just take that and tell you, okay, this tumor sample is probably a medulloblastoma or whatever, something like that. Does that make sense? Those who want to be doctors, are you excited? That's cool, right? And of course, these technologies are now old news. They're at least 5-6 years old. So things are getting better and better. Right? All right. What else can you do? You can do many more type of diagnostics and tests and etc. One of the things you can do is, medicine is some medicine. So in medicine, one of the problems is this. You have a treatment for a certain type of disease, but it doesn't work for everyone, right? Some people respond to the treatment and some people don't. wouldn't it be nice if you knew who's going to respond and who's going to not respond, so you don't actually use that kind of treatment on the people you know are not going to respond, right? So, for example, what you can do is here, I forget what kind of disease this is, but it's looking for the gene expression profiles for a bunch of people. So each column here is a person, and these people were responders and these people are non-responders, and they look at the expression profile of a bunch of genes, and this here they look at thousands of genes, but they're showing you only those genes that actually differentiate these two groups, okay? So when you look at this, what you can see is that these gene groups, this gene here, this set of genes here are highly expressed in the non-responders and lowly expressed in the responders. And these set of genes are expressed in the responders and not the non-responders. Then you can ask, which of these genes are the most reliable genes to tell me if that group is a responder of a non-responder? So what you can do is you can develop PCR assays for the 14 most diagnostic genes and determine if your patient is going to respond or not to the therapy before you actually go through the therapy, right? All right. This can be also used in research for actually developing drugs to certain therapies. So one of the drug therapy problems is this. You develop a drug, you treat a patient, you put it on cells, you put it in the mouse, you put it in the patient. If the drug kills everything, that's not a good drug, right? You want to kill cancer cells, you don't want to kill something else. So a lot of the drugs are toxic. So how can you test if a drug is toxic before actually injecting into people? You want to do these experiments in vitro. That means outside the organism, right? So you want to do these in dishes. You want to predict if a drug is going to be toxic or not. What can you do? What you can do here is, for example, you have a new drug candidate and you want to know if that's going to be toxic to liver cells. What you can do is you can grow liver cells in dishes, and you can add to these cells different toxins that you know are toxic to liver, right? And then you can add to these cells different drugs that you know are not toxic to the liver. So each of these rows is one of these drugs that you're adding, and each of these columns is the genes that are responding to the drug, right? And now you can take the gene expression data and cluster it and find those genes that are responding to non-toxic substances differently from toxic substances, right? And then you look at your experimental drug candidate and compare which of these groups does it belong to. So in this example, you have these genes all red and red, and these genes are green and green. Would you push this drug to the next step of the drug development? Probably not, right? Because you predict that it's already pretty toxic, and you predict that it's going to be toxic. All right. So that's microarrays, and microarrays are awesome. It totally changed the way people do business in both research and in medicine. And that technology is about 15, 20 years old now, and it's been pretty popular. But now a new technology is taking over a lot of these kind of diagnostics. It's not there in the clinic yet, but it's coming up. And that is, instead of microarrays, using RNA-seq to make these measurements, okay? So they're switching to high-throughput sequencing. We ran through some of these technologies to measure RNA abundance. So I'm just going to give you one example in which you can measure RNA abundance. We're going to talk about mRNAs again, not the functional RNAs like microRNAs and other things, though you can do sequencing to measure the amount of these type of RNAs as well. I'm just going to give you the mRNA because that's the easiest to understand. So first what you're going to do is you're going to isolate RNA from your sample. Then you're going to, if you want to look at just mRNAs and nothing else, you can actually isolate your mRNAs. And the way you can do that is if you use a oligo-DT, which is going to bind to the poly A tail, And that oligo-D2 is chemically cross-linked. It's linked to a bead. Okay? These kind of beads, you can buy several kinds of them. One of them could be magnetic. That is, if you actually have a magnet here, right? And you put your tube, you have all your RNAs in your tube. Some of them are going to be polyadenylated, and some of them are not going to be polyadenylated. What type of RNA is not polyadenylated and very, very abundant in the cell? I mean, there's like 95% of the RNAs in the cell are not polyadenylated. tRNAs, rRNAs, microRNAs, right, etc., etc. So what you want to do is you want to isolate the mRNA away from them. So what you can do is you can use a magnetic stand, and everything that has that magnetic bead with the poly-T that binds to this, what we'll do is they're going to go and stick to the side where you have the magnetic stand, and you can wash away everything else, so you can isolate your RNA that you are interested in, right? Does that make sense? Okay. So now you have your mRNAs. You want to make, the next step is to use random hexamers as primers once again. And you can use the first strand cDNA synthesis with the reverse transcriptase enzyme. Now, that first strand is going to be, when it's synthesized, you're going to have a DNA-RNA hybrid, right? The DNA is in blue, the RNA is in red. So that's the first cDNA. And then what you can do is you can use a collection of enzymes to create the second strand of the cDNA. So you make a copy of the RNA as a double-stranded DNA. And some of the enzymes that you use is RNase H, which cuts and makes nicks in the RNA portion of the DNA-RNA duplex. Okay? And then DNA polymerase 1 can use these little RNA pieces that are left from RNase H as RNase H as a primer to start making the complementary DNA. And the DNA ligase is going to start ligating whatever fragments that the DNA polymerase have synthesized. So at the end, what you're going to have is a double-stranded cDNA. Does that make sense? Any questions?
What happens to the uracil in mRNA? How do I
What happens to what?
to the uracil in RNA? How do we convert it to time?
I couldn't hear it.
What happens to the uracil in RNA?
Oh, uracil. So this DNA polymerase, whenever it sees a uracil, it adds a against it. Right? So it adds the correct nucleotide. So in the first strand synthesis, all your uracils have the correct.
But I mean when we ligate the red one.
Oh, these are all removed. You don't ligate these. So these little things. So basically, imagine like this. So you have your DNA and you have little RNA pieces. So RNase is gonna cut it, right? So these are going to be RNA pieces. And the DNA polymerase is going to come over here, and it's going to synthesize. And then when it hits the next one, it actually removes it out. And the RNase is also constantly chewing up more and more in the reaction. So it's going to go finish. And if there was another polymerase, that polymerase can only go in one direction. So if there is another polymerase that hooked up here, it's going to add that stuff. And when this comes here, this RNA is going to be removed, just like how Okazaki fragments kind of work, right? And the ligase is going to ligate here. So then you're going to have the second strand. Does that make sense? All right. So at the end, what you have is a double-stranded DNA. We talked already about how you can convert a double-stranded DNA into an Illumina sequencing library in the ChIP-seq example. So this part is exactly the same. once you have a double-stranded DNA, you can add an A nucleotide at the end using an enzyme called hexaclino. Basically, it is a clonal fragment, is a DNA polymerase from E. coli that lacks a portion of the N-terminal region of the protein that has nuclease activity. And that's why it's called exo clino. And then what you do is you ligate your Illumina adapters with a T overhang to these samples, and then you send it to sequencing to Illumina, and you get sequences. And the readout is this. What you're going to get from your sample is a whole bunch of sequences, and there are millions and millions of them. And the principle is this. In microRNAs, if you had initial RNA amount, right, if your initial RNA was a lot more, it's going to have a lot more fluorescence. In this RNA-seq, if your initial mRNA is a lot more, you're going to have more reads coming up from that RNA, right? Compared to another gene that has less reads. Does that make sense? All right. So here's an example. What you do is you take your reads, just like in the ChIP-seq example, right? You take the reads from your RNA-seq data, and you go to the genome and ask, where does this read coming from in the genome, right? And then you align your read to the genome. So this is a UCSC genome browser of the data. So what you're looking at here is you're looking at a gene. Again, the thicker rectangles are showing you the exons. The little lines are the introns. So in this gene, this is the start, exon, exon, intron, exon, exon. Each of these red rectangles is a read. So that's a 50 base pair or 35 base pair read sequence. So these reads basically aligned to this. That read aligned here, that read aligned here, etc., etc. These reads are a little bit curious, right? A portion of the read aligned to this region of the exon, and the remaining portion of that read aligned to the next exon. So can you tell me why this happened? Splicing. Because your cDNA is copying the DNA from the mRNA, and mRNAs are spliced. So when you have that copied, you basically have the sequence from here and here, but it's going to span through this intron. Would this happen in ChIP-seq? No, because you're starting with the DNA, right? There is no that mRNA converting into cDNA. What you do is you sequence DNA. So when you align your sequences back to the genome, it should match perfectly. It should not matter where the intron is. Is this clear to everyone? Okay. So here is an example from one of my students. I asked them, put all the reads on the UCSC browser and give me a shot, and that's what he gave. So these are C. elegans genes up here. So each of these is a gene. Again, the thick ones are exons, and the lines are introns. And these are all the RNA-seq reads that map to this region of the genome. And what you can see is you can see all kinds of reads. And the color coding here is if it's blue, it's mapping to the top strand or the bottom strand. So the sequence is the same as the top strand or the bottom strand, right? Because sequences have direction. And then from this you can see, top strand or the bottom strand. It's not that important. But one thing that I would like you to appreciate here is to look at this data and immediately tell me which one of these genes were more highly expressed compared to others. For example, tell me, in this sample, was the gene 1 more expressed or gene 2? 1. Perfect. Yep, so that's the principle. But that's not where we want to leave this, right? We just don't want to look at one gene and say, okay, this is more expressed. We want to quantify it. We want to turn these into numbers. How do we do that? We talked about turning these into numbers in microarrays using fluorescence signal that the scanner is getting, right? In the RNA-seq, the idea is very, very same. But what you do is instead of fluorescence intensity signal, you actually use read number, right? So you can have your treatment and the control, and for each gene you can count the number of reads that are aligning to that gene. And then what you can do is you can take a ratio of this and do exactly the same type of analysis in microarrays. So you can take that ratio, you can log transform it, and then you can color code it, and you can do clusters and any type of analysis that you want. Does that make sense? All right. Let's think about what is being measured here, right, between microarrays and RNA-seq. So what kind of information can RNA-seq provide but not microarrays for a given sample? Remember, in the microarrays, we make the microarray, right? We make a glass slide, and we put probes, which are DNA segments that are short but complementary to the genes that we are going to measure, right? For a given gene, I take a portion of that gene and I say, let me put that on the glass slide, and if that gene is expressed, it's going to hybridize there. In RNA-seq, what are we doing? We're taking all the RNA and we're making cDNA from the whole RNA. Then we're reading the sequences and going back and matching that into the genome. So what's the key difference here? I kind of told you. What can you measure with RNA-seq?
You could find previously unknown splice variants.
You can find previously unknown splice variants. I would actually go one step above that. You can find previously unknown genes that are expressed, right? That makes mRNA. You can find previously unknown splice variants. What else can you do? What information RNA-seq gives you that is not taken from microarrays? RNA-seq actually gives you the sequence, right? And you can look at sequence variability. You can measure alleles, right? You can see which allele of a gene is expressed more than other, right? You can do a lot more type of work using that sequence of the RNA. With microarrays, what you can do is you can only analyze what you actually put on the microarray, right? The other thing is the microarrays are limited by how many spots you can fit in a slide. Is mRNA-seq limited by that? No. You're just taking a whole collection of RNAs and making cDNA, and you're reading that. All right. Do we actually need to know the genome sequence of a species before we can analyze its transcriptome? What's the transcriptome of a species? The set of all the mRNAs that species can make. If I wanted to know what the transcriptome of a, give me an animal that you like. Zebrafish? Yeah, I'll take zebrafish. I'll probably want to take a whole bunch of them, eggs and etc. I'll probably want to grind it, right? And then I probably want to isolate the RNA from it. Zebrafish is that big, so don't worry. They're not that cute. No, they're a little bit cute. But anyway, doesn't matter. You can take that, you can get the RNA, and you can sequence it, and you have sequence information for all the genes. Do I need a prior genome sequence to analyze the RNA-seq data? Nope. In our examples, we did, right? Because you can align it back to the gene, and you can know, okay, which gene is expressed, etc. But even if you didn't have the genome, you will be able to generate gene models because of those reads that span the introns. You won't know what the intron sequence is, right? But you will know, okay, that RNA is basically this gene, and if you have different splice variants, you can guess where the exons are, right? So this is a great technique to study the transcriptome of animals who is genomes are not sequenced. So you can still study gene expression without knowing what the sequence is. Is that cool? All right. So both microRNAs and mRNA-seq that I showed you is a population assay. Remember, I told you about what a population assay is with ChIP-seq. That means you need multiple cells to look at that. You look at the average expression of RNAs in a collection of cells that is coming from your sample, right? So, for example, if you are a cancer researcher and you're looking at cancer, at a tumor, one of the things the cancer researchers are pretty struck with is that that tumor contains multiple cell types, right? But when you cut a tumor, you can't necessarily know that each cell is different from each other, and you can't necessarily separate them before you do a transcriptome, or you do a microRNA or a microarray or something like that, right? But you know that it's a mixture of cancer cells, normal cells, and cancer cells have different types. So when you do a microarray, you take an average of all that. Also, if you're working with an animal and studying its development as a researcher, for example, you know that in an embryo, each of those cells are destined to be something else, right? And if you can't dissociate those cells and isolate all the intestine cells from an embryo, if you do a microarray from those embryos, it's going to be a mixture of all those cell types, right? So then you're not going to be able to know which gene is being expressed in the intestine cells in a very early embryo. It's hard to get that transcriptome. So to study these type of questions, there are methods, right? There are methods you can use. One of them is single-cell RNA-seq. But before we move on to single-cell RNA-seq, I just want to show you some pictures. So this, we already know that in a given embryo or a cancer or whatever, or a developed tissue, we know that there are several cell types. And the reason we know that is that we already know some genes that are differentially expressed between these, right? So what you can do is you can visualize the different cell types based on what you know using transgenic. So here, what the researchers have done is they took their protein that they know are expressed differently in the different cell types, right? And they fused GFP, RFP, whatever that blue fluorescent protein was, the name, blue fluorescent protein, or all these different type of proteins that fluorescent at different colors, right? And then you put that in all these different proteins that are expressed differently and create a transgenic animal that's expressing all of those, right? Then you visualize these animals under a microscope. What you can see here, again, zebrafish, our favorite now. The zebrafish embryos, you can see that there are some cells expressing this protein, others expressing the blue, expressing the red, etc., right? And you can do the same thing with mouse lymph node where you can visualize different cells. The trouble is same as southern blot or in situ hybridization. You can look at a few a few proteins at a time, right? But you can look at a few genes at a time, and it's a lot of work to make transgenics. If you want to do it in a high throughput manner, you need another assay. Oh, before we go with that, let me ask you this question. How can you fuse the GFP coding sequence to your gene of interest? This is all about previous lectures. If you were going to do this, right, you're going to do this experiment in a mouse. You're going to tag a protein of your interest with GFP. What do you need to do first? You're going to build a plasmid. Don't hide.
With restriction enzymes that have complementary ends to the GFP.
Yeah, you can make a PCR primers with restriction enzymes and amplify the GFP. You have your plasmid, you clone your gene in there. You put the GFP right at the 3' of your protein. You remove the stop codon of the protein so they are fused, so that the ribosome is going to translate the whole thing. It's going to make your protein, and it's going to fuse it to the GFP. Does that make sense to everyone? All right. Now you're going to take that plasmid. It's going to have some selection marker, right? You're going to make a mouse transgenic, the way that Professor Siegel actually explained it, how to do this. You're going to select for the right mouse, and you're going to grow it, and you're going to put it under microscope and have fun. All right. So what we need is a high throughput approach to study this. One of the ways to do this is say, okay, I'm just going to step back, and I'm going to see if I can make RNA from single cell and measure its transcriptome. So I'm going to look at all the mRNAs of a single cell. So that is what is called single-cell RNA-seq. And the type of information you can get from single-cell RNA-seq is you can look at cell-to-cell variation of the transcriptome, right? And you can look at how that affects development and disease, such as cancer is a really good example for that. Here's how people can do this. So this is for neuro majors here. You might like this. I'm not a neuro person, but I'll do my best. So apparently there were these several regions in the brain that were known to have certain functions, and they were known to have certain types of certain cell types, right? So what they did is they took a 51-year-old female that died from natural causes and took brain samples from this person. And they took the brain samples and they isolated the nuclei separately from each of these regions. So when you take a brain sample, it's not just neuronal cells, it's some other cell types as well. So they said, we only want those nuclei that are coming from neuronal cells. And the way you can do that is if you have a neuronal marker, that means if there is a protein or something that you can actually separate the cells based on using fluorescence activated cell sorter, I think this stands for FACS. What you can do is you can only take those neuronal cells and leave all the other connective tissue cells or muscle cells, etc. out, right? So you take these, they took this, and then what they did is they put it into a newly developed machine called C1 capture that uses this technique called C1 capture, which is basically a microfluidics device that can separate each nuclei from each other into a different compartment so that you can do all the molecular biology in a very tiny compartment for one nuclei at a time. There are other methods based on this kind of single-cell separation, one of which is called drop-seq, and I know at least one of you is in a lab doing this right now. So basically what it allows you to do is allows you to take one nuclei and do all the molecular biology on that one nuclei and make the mRNA. Then what you do is you take those mRNAs, you sequence them, and you take the reads from each of these single cells and you map it back to the genome. Because this technology is a little bit hard, there's a lot of inefficiencies in it. So one thing that you can do is you can look how many of my reads are actually coming from exons, right? If you're actually doing an mRNA-seq, those should be coming mostly from the exons and not intergenic regions. Intergenic regions means regions between genes, right? And we know that most of those are not sequenced and polyadenylated. Not transcribed and polyadenylated. So what you can see is in this sample, a lot of the sequences were mapping to the exonic regions, which is kind of nice. Some of them were intergenic. That's okay. We know some intergenic regions are actually transcribed, but some of this is actually just background. That means it's just garbage, right? It's not correct. And some of them are mapping to introns because these are nuclei, right? They isolated nuclei. The transcription and splicing is going on. So there are some RNA species there that are just polyadenylated, but still they have their introns not just been removed yet. So you can capture some of the introns as well. And then what you do is you clean up your reads based on a bunch of parameters that throws away low quality reads. And now what they ended up with is they ended up with 3,227 data sets. So each data set is corresponding to one cell. Okay? What do you do with this? So far, we've seen clustering, right? And the principle between clustering is you take one sample and you say, which of the other samples resemble to this one sample I took? And you take that sample and put it next to it. Okay? So that's basically what it is. And then you say, which of other samples most resemble to this, etc., etc. And what that allows you to is group the samples, right? That's what clustering was. In this example, they are doing exactly the same thing, but what they're doing is they are grouping the cells in two somewhat arbitrary dimensions. This is just to be able to visualize this. Instead of a cluster, you're visualizing in a surface, okay? So what they did then is they color-coded the regions in which these cells are coming from, right? And what you can see is that the cells coming from the blue region of the brain, front something, you can help me later, but those cells are clustering together. What that means is those cells have a more similar transcriptome to each other than cells coming from the green region of the brain, and those cells look different from the orange region of the brain, etc., etc. And what they found is that within a region of the brain, there are also groups of cells that have more similar transcriptomes. That means within that region of the brain, there were other cell types that the researchers are not yet aware of, right? There are certain subtypes under each cell type, right? So, okay, so they look at 6 distinct regions of the cerebral cortex, and this will give them a kind of a nice transcriptome atlas to go back and look at the brains of other people or study the different cell types and see if they have different functions, right? Some of these cell types or subcell types in a given region might have different functions because they're expressing different genes. Does that make sense? Is that awesome? Yeah, pretty cool. All right, I think that's it, and I'll see you on Monday.